Okay, the allele frequency lab. Okay, so first, gene pool is all of the alleles that are currently existing inside a population. Okay, and so we have two different alleles. The colored bead represents the A allele, which in America is 100% a healthy hemoglobin, and then the white one represents the S version of the hemoglobin. It is the S allele. An allele is a version of a gene, and so it causes a version that cannot properly bind to oxygen, and so not being able to get oxygen in your body is a big deal. All right, so what we did was you pick two beads at a time, because even though alleles exist, what actually interacts with the environment is always a genotype. Alleles do not interact with the environment, only a genotype does. So that's why it's still in the cup. It's not interacting with the environment. Okay, so you pick two beads, you pull them out, and now boom, I need to interact with the environment. So I have an AA genotype person, and so in America, this survives 100% of the time because there is nothing, it just does. It's a healthy, two healthy hemoglobins, you're getting oxygen to your body. So I put it in that cup. All right, so another AA, I'm gonna cheat and actually look. And so in this case, I get a heterozygous genotype, which means I have two separate copies of the allele. And so this, even though they are called a carrier, meaning they have the allele, it's there, they carry the allele, but they don't actually have the gene, because even if you have one copy of a healthy hemoglobin, you're able to get enough oxygen to keep you alive. Okay? What ends up happening though is, and by the way, this isn't true in modern world. We have medicines to keep someone alive who has the SS genotype. But historically, what would happen is the SS genotype would end up being lethal. They end up sticking together because it creates a nonpolar residue on the outside, and they stick together and they cause the cells to sickle, and then the cells get stuck and cause death downstream of it. So anyway, so what ends up happening is the SS doesn't continue to exist. It actually ends up dying off forever. Okay, so I put those there, which means two things I'm going to do. Number one, I'm going to go ahead and flip the SS cup because this person never survives, and then I'm going to go ahead and move these back because they're in the ground. All right, so this isn't a possibility because SS die, and then they're no longer in the gene pool generation because they can't. And so anytime I would pick an SS, I would move them back, and so the ones that will keep going will be here. Okay, and so I'm going to keep picking, and so let's imagine that what ended up happening was I picked two more sets of SS's. Those both would die. Okay, and then, right, so that's one here, and most of them would be here. All right, and so what really is happening is these are still part of the gene pool, but this is the gene pool of the next generation. Okay, and so the gene pool is all the people who pass on their genes. So if you're a person like Casey Jones who doesn't have kids, all of your genes actually can go ahead and be deposited in the non-surviving genes anyways, because your genes only survive if you have offspring. Okay, and so yeah, so that's what goes on. All right, so what we then do is we pull them all back into the gene pool, and then when you pick them again, what ends up happening is this is then the next generation. So whenever you start an experiment keeping track of alleles and genotypes, what you do, the first generation is referred to as the P generation. The generation I just completed was the F1, and then this now would be the F2 generation. And so for all of you, when you meet your grandkids for the first time, you'll be like, oh, it's so nice to meet the first member of my F2 generation. I know you would never say that, but you should. All right, so what this is actually an example of is something called directional selection. So what is happening is, since one version of the allele, one genotype, which obviously causes a certain phenotype, is dying off, the population is actually moving in a direction because this particular gene pool is changing. All right, and so in this case, what would eventually happen over a long enough period of time, all of the S alleles would die off, and that's why the S genotype is so rare in the United States. All right, so what was the wrinkle though? The reason why the sickle cell gene is so high in certain parts of Africa and also in certain parts of South America, basically along the equator, is there is a mosquito that carries, and so the mosquito is a vector. It's not the mosquito that makes you sick. The mosquito has a parasite that when it bites you, the parasite gets inside of you, and that parasite causes a disease called malaria. All right? So the mosquito is a vector because it just carries the thing that makes you sick, even though itself. And so the malaria could never get inside of you, or would almost never be able to get inside of you if it wasn't for a mosquito breaking your skin, because we all know how important your skin is, right? So, anyways, so what ends up happening is in this case, there's something called a heterozygous advantage, because if you have one A and one S, the A is keeping you healthy by having a healthy hemoglobin, but this S does something to your blood, which still isn't completely understood, which makes you resistant to the malaria. Doesn't make you resistant to mosquito bites, you're still getting bit by a mosquito, but the malaria parasite, if it gets transferred to you, isn't able to survive thanks to this. So this actually is what's called a heterozygous advantage, because this one always survives. So what ended up happening is if you end up being born AA, you will survive when you're born, but what you did was you flipped this to see what is the probability of getting bit by a mosquito that has malaria. And so again, this is a historical representation. We have anti-malaria drugs which keep you alive today, and so if you go to countries that have it, you actually take these drugs with you, or you can in some cases even get vaccines. And so what happens is, again, it's a matter of the genotype, not an individual allele interacting with this, right? So this is your genes, and then this is signifying, boom, do I get bit by a mosquito? If you do get bit by a mosquito, oh well, you got malaria and you are now non-surviving. So that is a heterozygous advantage because the survival of the heterozygous is higher than either of the other possible homozygous dominant or homozygous recessive trait. That is allele frequency. Thank you for your attention and good luck on the AP exam.